All right. How are you guys? Welcome to Zoom Boost. So today we're talking about embryology, right? The fundamental embryology, the beginning of everything. Okay. So that is our discussion for the day. So we're going to be looking at the embryology from week 1 up to week 3. Okay. From week 1 up to week 3. And we should be familiar that the first week is counted from the day of conception, that is the day of fertilization of the ovum, to the day of implantation. And we know implantation starts at approximately day 6 and day 7 and it ends at day 13. Okay, so that is our week 1. So it's from ovulation to implantation. All right, so basically from ovulation, if fertilization happens, then to implantation. All right. Okay, so basically we have to go back and see how we end up having germ cells. So these germ cells are the ones that we find within the gonads, right? So the germ cells are the ones that we find within the gonads, right? The germ cells are the ones that we find within the gonad, and within the gonads, that's when we have spermatogenesis and oogenesis occurring. Okay, so we'll be having these germ cells, right? And within the testes, we have the spermatogonia, and then within the ovum, we have the oogonia. So we want to see where exactly they come from. So, this is an embryo over there, right? That's an embryo over there, right? So what happens is that these primordial germ cells, they migrate from the epiblast layer, right? Epiblast layer, and they come to rest within the wall of the yolk sac, right? They come to rest within the wall of the yolk sac. That is around the 3rd week, right, of development. Then they travel, right? They travel to the gonads, right? They travel to the gonads, and then reaching the gonads by the 5th week, reaching the gonads by the 5th week. Then by the 6th week, they actually enter the gonads, right? Actually enter the gonads. And then they cause now the differentiation of the gonads into either the testes or the ovary. And you understand, even from your reproductive physiology, that before the 6th week, we have the indifferent gonads. That means the gonads cannot be differentiated into either the male gonad or the female gonad. So they'll be waiting for these primordial germ cells, right? These primordial germ cells. Okay, fine. So that's basically it. So we say from epiblast to the wall of the yolk sac, from the wall of the yolk sac to the gonadal ridges, right? Fine. Now let's look at the overview of oogenesis. So there is this germ cell, right? There is this germ cell that called the oogonia, right? So within the ovary, the oogonia will find the oogonia within the cortex of the ovary, all right? Within the cortex of the ovary, within the medulla of the ovary, we find the neurovascular structures within there. So find the neurovascular structures within the medulla of the ovary. So they are found within the cortex. All right. Fine. Why? Because the gonadal ridge, all right, the indifferent gonad, right, has got a cortex in the medulla. And then in the female is the cortex that forms the ovary. And in the males is the medulla that forms the testes. So there we have the oogonium, right? So it's diploid, as we see. It's actually diploid, right? So it will actually then divide there to form the primary oocyte, right? So this division is like mitosis, or we can call that what? Mitosis there, right? So thereby mitosis, there we have the primary oocyte, okay? And then now we start the process of having the number, right? The chromosome number, right? To the secondary oocyte, and then from the secondary oocyte, we then divide again, right? Like that. All right. Okay. All right. So in the process of this division, right? In the process of the division, the other cell that comes out has got less cytoplasm. So there's an equal sharing of the cytoplasm, so that the first polar body is given out and non-functional because it has less cytoplasm. So that's the first polar body. And then the second polar body will be produced, the second polar body will actually be produced at fertilization, right? Because at fertilization, that's when we have a complete division of the secondary oocyte. That's when you have a complete division of the secondary oocyte, right? So if I can actually label very well, though this division is like mitosis, but I really want to call that meiosis 1. And then we have meiosis 2 over there, right? Right. So that is the principle that we have there, okay? That is exactly what we will be having over there. All right. So then at last we have our ovum, right? That will then be released and be fertilized, right? But now the process of oogenesis takes a lifetime, different from the process of spermatogenesis that takes approximately 72 to 74 days, right? 72 to 74 days. But for the oogenesis, it starts within the prenatal life that is within the womb, right? And then ends at fertilization, right? So actually what we see is that by this division, what we actually see is that we, at birth, so that's, let me just put this line to indicate at birth, right? So at birth, what we actually have is our egg that is at the level of prophase 1. So that will be at the level of prophase 1, and then they rest over there, right? And we'll be calling them the primordial follicles, right? And then when one is born, they are still at prophase 1, right? And then one of these cells at prophase 1 will be selected every month, right, to complete the cycle, right? And then in completing the cycle, they will be rested at metaphase 2, right? They'll be rested at metaphase 2, right? And then as long as now they are fertilized, so as long as they are now fertilized, what you actually see is that they can now complete metaphase 2 and come out as the ovum, the mature ovum, right? And the second polar body, and the second polar body, and the second polar body. So at the end of all this division, we see that one oogonium will give 3 polar bodies. How? This is, let's count that number 1. And this polar body will actually divide to form other 2 smaller polar bodies. So we call that 1, 2, and then 3. So we form 3 polar bodies, right? And 1 mature ovum. All right, 1 mature ovum, right? So we start by rapid division of this oogonium, right? Rapid division of this oogonium, and then halving of the chromosome number over there. So halving of the chromosome number over there. All right. So then we go on to spermatogenesis that we say it takes approximately 72 days to be finished. So we start as well with the spermatogonium that are located within the seminiferous tubules. But if you look at the seminiferous tubule, what actually happens, what you actually see is that the tubule has a lumen, right? And then it has a basal compartment. So within the basal compartment, that's where we have these spermatogonia. So they differentiate and divide, moving towards the lumen. And then the final stage where they're given the tail and then they're reduced the amount of cytoplasm, which is called spermiogenesis, occurs on the adluminal compartment that is closer to the lumen. So called the adluminal compartment. And then in their division, there are supporting cells that we call the satellite cells. That we call the satellite cells. So these satellite cells, what they do is that they nurture these cells, right? Also creating what we call a blood testis barrier. Also create what we call a blood testis barrier. So these are the satellite cells. Okay. satellite cells in nurturing these dividing germ cells, right? And then outside that, we find that within the interstitial between the one seminucleus tube and the other, we have another hormone-secreting cells that are called the Latic cells. So the Latic cells are hormone-secreting cells. So called that the Latic cells. So they produce the testosterone, and then that testosterone is transferred to these dividing cells, right? Transferred to these dividing cells, and also that testosterone also enters the vascular system, and is transported to different parts of the body. All right. There is a feedback as well to the brain for the production of FSH. Okay. Right. So that's basically that. So there we have these ones in the basal compartment, and then now we're moving towards the lumen. Right. So the first division is whereby we have mitosis, right? So the first division is there, we have mitosis, right? So that is rapid division of the cell without any differentiation occurring, right? So here we generally have multiplication, right? And then lastly, we then have what we call a primary spermatocyte, and then this will divide by first meiotic division into secondary spermatocyte, right? And then the second meiotic division into these, what we call the spermatids. And the spermatids will then be given the tail and be reduced in cytoplasm and be given the acrosome, right? The acrosome, and then become the spermatozoa. So what I want you to remember is that mitosis is that process that to form the primary spermatocyte. So the primary spermatocyte is diploid. The secondary spermatocyte is haploid, right? Everything, right? So as well, if we come back, let me look here, that was supposed to be mitosis. Just right? And then first meiotic division, right? First meiotic division, it's actually there now, we're correcting ourselves. First meiotic, then this is now second meiotic division. It makes a lot of sense now. Second meiotic division, right? Now, so there we are. So I want you to understand that when we divide the spermatogonia, we get two types of cells. Generally, the spermatogonia, when they divide, they form two generations. There is the darker cells that we call the type A, and then we have the light cells that we call the type B, right? So the darker cells are called the type A, and the lighter cells are called the type B. What I actually see is the type B is the one that will continue the journey going downwards, up to we form the spermatozoa. So it's the type B or the light type, right, that will continue the journey of dividing, or the journey of spermatogenesis. What I actually see is the type A has got a recycling effect. The type A has a recycling effect. So the type A will continue dividing, right? Continue dividing, right? Continue dividing, right? Continue dividing, but dividing by mitosis to restore the spermatogonia. So it actually divides to restore all these diploid cells, right? So that one won't have like a deficiency even if they age, right? They will always be having this spermatogonia. So they don't reduce in number, right? They don't reduce in number. It's different from the females. The females make all of their primary oocytes, right, during prenatal life. So they reduce by age. Right? For the males, because of this type A cells, they quickly divide by mitosis, regenerating back. So there's always that cycle occurring. But the type B, when they form, they go home. Type A, when they form, they regenerate back. Right? And the last stage when we form these spermatids over there. So here, what we actually have is cell division, right? Cell division. And then lastly, what we have is differentiation, which is called spermiogenesis. So spermiogenesis, what we're actually doing is that we're reducing the cytoplasm, the amount of cytoplasm, right? So if that is the cell, the cellular cells, what actually do is that they'll come in and they'll eat, they'll chunk this part. So they're actually like macrophages, actually come in and chunk that part, so they'll eat that part, right? And then that part is removed as what we call the residual body. That part will be removed as a residual body, and then they will now have the spermatozoa. Maybe that is done for movement, right? So one is bullets these bullet-shaped structures of the thing, right? That are fast moving, and then they're given the tail, right? So all of this is done by the mothers, which are the satellite cells, right? So the satellite cells, their structure, they span from the basal compartment to the adluminal compartment. These are long cells. So they'll span the whole length. They'll span the whole length. And then you have the nucleus over there. So these are just tau cells, right? And these cells now have got set gap junctions. So they actually share their cytoplasm with the dividing cells, right? So they do a lot of nurturing. And then we also talked about how these cells have a function in the blood testis body, forming the blood testis body, all right? And then also some of the functions of the satellite cells is information of what we call the androgen binding protein, informing what we call the androgen binding protein, right? Androgen binding protein. And then also in development, they also form what we call the Mullerian inhibiting substance. The Mullerian inhibiting substance that will remove the cord that is supposed to make the uterus in the males. That's why the males don't have the uterus as well, right? So that is produced as a chemical that is called the Mullerian inhibiting substance that is produced by the satellite cell, right? Anyway, so now the process of ovulation now. So ovulation occurs on day 14. So ovulation occurring on day 14, right? By the surging of the luteinizing hormone. So the surging of the luteinizing hormone, right, will induce ovulation, right? So when one is born, they have these, what we call the primordial follicles. So they have these primordial follicles. The primordial follicles. And the structure of the primordial follicle is one cell, right, the oocyte, that is surrounded by these squamous cells that are called the follicular cells. That are called the follicular cell. And then from this primordial follicle, we go on to the primary follicle when the cells are now cuboidal, and then to the secondary follicle when these cells are now these follicular cells are now forming a strata around, right, up to we reach what we call the Graphene follicle. But for now, I'll talk about the Graphene follicle. So just before ovulation, what we have is this mature Graphene follicle, right? So the structure of the Graphene follicle, so these cells that are called the follicular cells, they will differentiate and become columnar, become cuboidal, and then form this strata over there. So it actually looks like a stratified cuboidal, right? Stratified cuboidal over there. So there we have the oocyte over there. And these cells are forming the stratified cuboidal. And now separating these cells and the oocyte is a thick layer that we call the zona pellucida. That we call the zona pellucida. And then now these cells now are now called granulosa cells. So the cells are now called granulosa cells. And you see that between the cells is a fluid that we call the antral fluid. So we call this the antral fluid. So this space is called an antrum. So the fluid within is called antral fluid. It has lots and lots of hormones produced by the granulosa cells. So these are also hormone secreting cells. Now, interestingly, is that outside the granulosa cells, we have cells that come from the tissue of the ovary, and then they just thicken around. So they're called the thicker. So they're called the thicker. So just thicken around. But these thicker now are divided into two. So there's thicker externa and then thicker interna. Now you can't see clearly. Let me just illustrate here. So you're having a thicker externa that is fibrous. So it's lots of collagen, lots and lots of collagen, and then few cells, and then also fibroblasts within. So if you remember the histology, it's almost like dense connective tissue irregular, right? And then within the thicker interna is cellular. Is actually cellular. Remember the pericondium and the periosteum, almost like that. So it's full of cells. Actually full of cells. So if a question comes and asks you which of these thicker layers has what functions in the production of hormones, you actually now know it's the thicker interna. So this one has protective functions, thicker externa, which is fibrous, and the cellular is the one that produces hormones. So actually these cells produce androgens. And these androgens will be transferred, right? These androgens will be transferred to the granulosa cells, and the granulosa cells, right, will then use an enzyme called aromatase to form the female hormones. Okay, the female hormones, and then the female hormones will be transferred into the antral fluid and also have functions in the differentiation of the oocyte over there, right? So the estrogen, right, that's from there, right? So that's the basic structure, right? So take external and then take internal. So at ovulation, what you actually see is that the cells surrounding, so I can label it here, I can just cut over here and then say that these cells that I've created the boundary, these cells will be released as well with the oocyte, and the remaining cells will remain as well with the figure. So this part that will be removed, right, that's our egg now, right? And the part that will remain as the corpus luteum, right? The part that would actually remain as the corpus luteum. So let's talk a little bit about this corpus luteum. So at ovulation, we now have a corpus luteum that is left, right? And the egg is gone. So we have ovulation within the peritoneal cavity there, right? And the egg is actually gone, right? So what is the function? What will actually happen? What is the fate of this corpus luteum? So if the egg is fertilized, I want you to listen clearly. So if the egg is fertilized, what we see is that the egg would differentiate into an embryoblast that will form the embryo, right? That will actually form the embryo, right? And it will divide into a trophoblast that will become the covering of the embryo. The trophoblast that will become the covering of the embryo, right? So I'm just represented by T. So the trophoblast that will become the covering of the embryo, right? The trophoblast that will become the covering of the embryo, right? So what is the deal now? So what I actually see is that the trophoblast will secrete hormones, right, that will maintain the corpus luteum. Right. So the trophoblast will secrete hormones that will maintain the corpus luteum, right? That will actually maintain the corpus luteum, right? These are the hormones of pregnancy, right? Right. And then this corpus luteum will then start producing progesterone as well as testosterone as well. But the main hormones that I want here is progesterone as well. Not testosterone, sorry. Let me just focus on this progesterone, right? As well as some amounts of estrogen as well, right? That's why we see that after ovulation, we have a high surging and high increase in the amount of progesterone, right? So what progesterone will actually do, that progesterone will then come to the uterus in the endometrium of the uterus. The endometrium of the uterus, and it will maintain the endometrium of the uterus. So it actually come and then maintain the endometrium of the uterus, preparing the uterus for implantation, so that the egg can then now come and implant, right, on suitable grounds. So you see the cycle. So we said, if this is fertilized, right, it will divide into an embryo and a trophoblast. Trophoblast secretes hormones that maintain the corpus luteum, and then that's called the corpus luteum of pregnancy. The corpus luteum then secretes the progesterone that will then maintain the uterus, the endometrium of the uterus. So that means everything is now waiting for fertilization. So what if we don't have the fertilization? What if we don't have the fertilization? Then we don't have the differentiation into the embryoblast and the trophoblast. And then we don't have the maintainer, right? And then now what I can see is that we don't have the corpus luteum being maintained, right? So we don't have the maintainer for the corpus luteum, right? So what we see is that the corpus luteum will actually degenerate, right? And then that's called the corpus albicans. And then when it degenerates now, we don't have what? Progesterone, right? So we have a fall in the amount of progesterone, just a quick fall at the end of the month. So if we have a quick fall, what actually happens? The maintenance of the endometrium will actually then stop. So what we have is then menstruation, okay, at the starting of the new month. All right, so that's the whole cycle. So anyway, that is about ovulation. Now let's talk about fertilization, right? So fertilization is a triple phase process, and I'll explain it all. So it occurs within the ampulla of the fallopian tube, the ampullary part of the fallopian tube. You mustn't forget this, very, very important. And then marks the end of meiosis 2 of the ovum, right? So remember we said meiosis 2 will be rested at metaphase 2. So at ovulation, we have these at metaphase 2. Right? So it will then be completed just after fertilization. But I'll talk about the process of fertilization when this will be completed, right? Now, the spermatozoa, right, that are inserted within the female genital take approximately 30 days to move from the cervix, 30 minutes, sorry, to move from the cervix to the ampulla, to that region, the ampular region, but can delay up to 6 days, right? And then in the process of moving, what is supposed to happen is that there should be a process that we call capacitation that will be happening there. So that is preparation, right? And the second reaction that will actually happen is acrosome reaction that will happen in the process of digging the zona pellucida then. So capacitation takes approximately 7 hours. So imagine, so if it moves very quickly and then takes 30 minutes to reach the ampulla, it won't fertilize until 7 hours is done. So even if it moves quickly, it has to wait for 7 hours, right? And then this process of capacitation occurs by what we call epithelial interaction between the head, right, of the spermatozoa and the epithelial lining of the fallopian tube. And that process is generally the removal of the glycoprotein layer, right, as well as the plasma membrane. Okay. Then the acrosome reaction now, when the spermatozoa is trying to dig deep to the zona pellucida, right? So say zona pellucida is that layer, right, of protein that is, of glycoprotein that is separating the oocyte from the granulosa cells around, right? That we call the corona radiata. Right? So after piercing the corona radiata, when this head of the spermatozoa now gets in contact with the zona pellucida, on the zona pellucida, there are proteins that we call the ZP proteins that act as receptors. So once they are in contact, right, they will induce the head of the spermatozoa, what we call the acrosome, that is the front of the head, to produce these digestive enzymes that will digest that part of the zona pellucida, right? So that is the movement. So we said from here to the ampulla takes 30 minutes or maybe later 6 days. In the process of movement, we have capacitation occurring in this process, right? Capacitation occurring. And then we have here the ovary, right? There we have the corpus luteum over there, right? And then here we have ovulation, right? Ovulation. And then this is the fimbrial part of the fallopian tube, right? And then move up to that point. All right. And then you have to understand that ovulation occurs within the peritoneal cavity. So this whole space is the peritoneal cavity, right? This whole space is the peritoneal cavity. So you understand ovulation occurs within the peritoneal cavity and then enters the tube. Okay. So it's not done exactly within the tube. Then now, so that is the process, the acrosome reaction. So that is the acrosome. right? So that's a bag, right? That's a bag. It's like a large lysosome, right? There's large lysosomes there, right? So what we see is that by interaction with the ZP proteins, there is what? Fusion of this acrosome, right? And the acrosome is actually based, right? Acrosome actually based, and in this enzyme will then digest the zona pellucida, digest the zona pellucida to allow what? To allow the entrance, of the nucleus inside, to allow the entrance of the nucleus as well, right? So anyway, let me just explain some of the processes what that occur there, right? So if we have outside, we have the zona pellucida there, right? That is the zona pellucida, and then inside we have, right, the ovum, right? And then we say outside that we have cells. So outside that we have cells, right, that are called the corona radiata, which is generally granulosa cells. The corona radiata is outside. So these are the layers that has to be pierced, right? Number one, the corona radiata. Number two, the zona pellucida, right, which is the major barrier. And then number three, the plasma membrane of the oocyte. All right. So what I actually see is that we have capacitation occurring, number one. To dig to this zona pellucida, right, we need the acrosome occurring, number two. And then the presence now of the nucleus, the male pronucleus, right? So there is the female nucleus, right, over there, right? So the presence, right, of this will induce the granules that will be within this cytoplasm, granules. So these small granules to actually come and fuse with the plasma membrane and release their contents, fuse with the plasma membrane and then release their contents there. And these contents actually harden. What these contents actually do, harden the zona pellucida. So the zona pellucida now becomes more hard. So that whatever spermatozoa is outside there and also trying to get in will fail, right? It will actually fail. And that is what functions in preventing what we call polyspermia, right? That is fertilization of one egg by more than two spermatozoa, right? So it will actually harden so that whatever, even if you reduce the enzymes, nothing happens, right? Okay. So that reaction is called the cortical reaction. That is reaction number three, right? Cortical reaction, that is reaction number three. All right. Then the last part of fertilization is then the fusion of this pronuclei. So that is the male pronuclei and the female pronuclei. So before they fuse, what you actually see is that there is condensation of their chromatin. So there's condensation of their chromatin. So if they so the chromatin has to condense, right? The chromatin has to condense, right? Actually has to condense, right? And then for us to be able to have pairing of the chromosomes. So it actually has to condense to have pairing of the chromosomes. Right. And that is where we have now the determination of the individual sex, either to become XY, which is the male, right, or XX, which is the female, right? Now, so at what point, what induces metaphase 2 to be completed? Are we supposed to have metaphase 2 completed after the fusion? No, it doesn't make sense, right? Because we first have to produce the polar body in the actual ovum. So the presence of this nucleus within the cytoplasm sends signals, right, that will induce this secondary oocyte to finish off metaphase 2. So the presence, right, not the fusion, the presence of this guy within that will then induce this one, right? Are we together? Fine. So then the process of implantation. So first we start with one cell, which is the zygote. One cell, which is the zygote, that is surrounded by what? The zona pellucida. So the zona pellucida is now thick, right, and resistant. So what we see is that the first part that we see is the division of this cell into two, right, and then division into four by mitosis, right, and then also divide as well, right, into 6, into 8, average given like that, right? And then that division now, right, is only the increase in number, but no increase in size. Why? Because the zona pellucida there is thick and resistant, right? Thick and resistant, right? So these cells can't expand, so they only increase in what? In number. So that's when they appear like a morula, right? Actually like a mulberry. So they actually appear like a morula. So these cells actually appear like a morula, right? And together, right? And these cells are what then what we call the blastomeres. So we call these cells the blastomeres. The blastomeres, right? So in the process, now in the journey to implant, moving from the ampulla to go to the uterine cavity, what we see is that there is fluid that will actually enter, right, through the zona pellucida and then enter into there, right? And this fluid, so at first we have now the blastomeres there, right? So the fluid will then enter, so this fluid that will accumulate over there, right? The fluid will actually accumulate over there, right? And in the process of this fluid coming to accumulate over there, this cell mass, this morula actually divide, right, into cells that will be outside. So these cells will divide themselves into outer cells and inner cells, right? Outer cells and then inner cells. All right, and then we say we have fluid there. So this is just illustrated on this little diagram. So these outer cells will form what we call the trophoblast. That will form the placenta. And in the inner cells, which is the embryoblast, that will form the human. Right? So there we are. The inner cell mass, the embryoblast, the outer cell mass, the trophoblast. This is the trophoblast that will say it actually secrete hormones, right? That will maintain corpus luteum, right? So there we have it. The inner cell mass, right? And then the trophoblast outside. And this is the trophoblast that has got functions in implantation, right? It is actually that trophoblast that has got functions in implantation, right? So what I actually see is that this trophoblast is the one that will dig deep, right, to the endometrium. So if the endometrium there, right, these trophoblast cells are the ones that will be digging deep, right, into the endometrium, right, until we have now fully embedded. And we say implantation starts at day 6 to end on day 13, right, on day 13. And what I actually see is that at day 13, the maternal blood vessels, maternal capillaries within the endometrium, they actually burst. They actually burst. So that bursting now will cause the release of blood, right? Which doesn't happen to all. This blood that you see here now being released, right, can be confused with menstrual blood, right? Can actually be confused with menstrual blood. So they can cause miscounting of the days there. Okay, right. All right. So that is the process of implantation. So day 13, that's when you have the plug, what we call the implantation plug, when it is now fully embedded within the endometrium, and then we have a plug closer, right? So there we have it, the inner cell mass, right? And then the outer cell mass that will form the trophoblast, right? The outer cell mass that will form the trophoblast, all right? Right. So the process of implantation, right, has to happen within the body of the uterus, right? The body of the uterus, right? Have it together. But the common position is here, right? Around the position that we call the fundus of the uterus. That is the common point. But we can have implantation occurring on the internal walls of the uterus, right? Within the fallopian tube. Now these are programs now, right? Within the fallopian tube. or within the ampulla, right? Or we can have it, we say it's released first in the peritoneal cavity. So it has to be sucked here, right? By the movement of the villi, right? Within the uterine tube. But it may actually move, get lost and then come on the walls of the small intestines or on the mesentery of the small intestines. So on the walls of the small intestines or the mesentery of the small intestines, it can actually happen with ectopic pregnancy. Or it can actually happen on the egg itself. That is, if the spermatozoa moves, right, and then comes over there. Now let's talk about second week now. We have fully implanted. So this is now what we call the week of twos, the second week. So first, our inner cell mass will then flatten to form a disc. So if you're looking from above, it will actually flatten to form a disc, right? But interestingly, this disc is in two layers. So this disc is actually in two layers. So if you're looking from sideways, this disc is actually two layers. Like two layers, right? I can divide it into two layers, right? An upper, what we call upper epiblast layer, and then a lower hypoblast layer. So that's easy, right? Lower hypoblast, and then an upper epiblast layer. Upper epiblast layer. So that is the important thing that happens at the second week, right? Okay, so that's number one. So now we've got two germ layers. So the first two germ layers, right? Done. What else is also true? Our trophoblast layer, which is covering the embryo, our trophoblast layer will divide into an inner cytotrophoblast, and an outer syncytial trophoblast. So this syncytial trophoblast is a bit like macrophagic in structure, right? So you see it is got funny shape, right? This syncytial trophoblast. So now we've got two. So the trophoblast is now divided into an inner cytotrophoblast and an outer syncytial trophoblast. So what is then the use? This cytotrophoblast generates the syncytial trophoblast, right? And one of the structures that we see in the syncytial trophoblast is that this syncytial trophoblast is multinucleated. So these are just the cells that fused, and now we see the nucleus all over. It's like one big thing that's called, it's like a cinctia. That's called the syncytial trophoblast. Actually, multinucleated structure. And it's got corrosive effects. So this is the one that will be corroding the maternal blood vessel. Is the one that will be corroding the maternal blood vessel, right? Maternal blood vessels. And also has got functions in forming the placenta, right? It functions in forming the placenta and also forming the barriers between the maternal blood and the embryo. So if I can draw the embryo, right? Maternal blood and the embryo. So that's when we have the first barrier that we have there, right? The first barrier that we have there, right? So it's got functions. So the trophoblast is the function in forming the placenta. So that is what that will form the other two. What else will be two as well? What else shall be two? You start to see that in association with the epiblast, there is a cavity that will form on top of the epiblast. There's a cavity that will form on top of the epiblast that we call the amniotic cavity. The amniotic cavity, right? And there's a cavity that will actually form below the hypoblast. There's a cavity that will actually form below the hypoblast, right? And that is called the yolk cavity. So there's two cavities there, right? So we call that the yolk cavity, right? So you really see that it's a week of twos, right? It's a week of twos. Then we say covering all of them is the trophoblast, and specifically the cytotrophoblast. The cytotrophoblast. So we say the epiblast, hypoblast, yolk sac, amniotic cavity, right? Or yolk cavity, amniotic cavity, right? The cytotrophoblast and the syncytotrophoblast outside, right? That is occurring in the second week, right? So there in the second week, we now have full embedding, and then we said that's the cord, right? Implantation plug. We have closed that plug, right? And then now we see what? The epiblast, that we said the epiblast are in association with this cavity. So there's a cavity there, right? That we call the amniotic cavity, right? And then now we have this yolk cavity, right? Yolk cavity. And then we said in association with the yolk cavity, we have the hypoblast cells, right? These cuboidal cells, the hypoblast cells. And these columnar cells are now our epiblast cells. Then outside that, we say we have now these cells, mononucleated cells that are the cytotrophoblast, which are in contact with the embryo. And then outside that, we have the syncytotrophoblast. Can you see it's all multinuclear? It's like one big structure that is many nuclei, right? And what you see is that it corrodes what? The blood vessels, right? It corrodes these blood vessels. And these blood vessels will actually then start leaking. And then what I actually see is that within the cytotrophoblast, we actually see spaces that are called the lacunae, spaces there. And this blood will come in and enter into this lacunae. This blood will then come and fill the lacunae, right? Fill the lacunae, fill the lacunae, fill the lacunae, right? But we won't see the lacunae, we won't see the lacunae within the cytotrophoblast, right? So that still creates the barrier over there. That still creates the barrier over there, right? Okay. So that what we see with time is that for us now to form the definitive barrier, right? The definitive barrier is that what we see is we will have what we call the extra embryonic mesoderm, right? Just outside. What we call the extra embryonic mesoderm. Extra embryonic mesoderm. So this is the extra embryonic mesoderm. Extra embryonic mesoderm. So what we then see is that we have these layers. So number one is the extra embryonic mesoderm. Number two is the cytotrophoblast. Number three is the syncytotrophoblast, right? And then number four is these blood vessels, right, within the cytotrophoblast. And the blood vessels within the cytotrophoblast. Actually, not say blood vessels, but blood. The blood vessel is outside the cytotrophoblast, but it can actually leak, right, its blood into there, right? So within the extra embryonic mesoderm, we also see the blood vessels forming from the cells of the embryo, right? So that's the barrier over there, right? So you see these layers are forming the barrier over there, right? Okay. And then from there, that's when we start to have what we call the villi, right? The villi, right? That won't be talked about in this video, right? What we call the villi, and the primary villi, the secondary villi, and the tertiary villi, right? But later on. So for now, let's just appreciate that these layers also have functions in forming the barrier between the maternal blood, right, and the blood of the embryo, right? Okay. Now, week 3, the week of threes as well, right? Or the week of gastrulation. The week of threes or the week of gastrulation. So what we see is that, so remember we have, at week 2, we have this disc, right? And then it's got two layers, epiblast and hypoblast, right? And then we say on top of it, so you see this, this gutter shape, right? Structure, this basin. That's when we have the amnion, and then below the hypoblast, we have the yolk. Okay. So week 3 is now the week of gastrulation. So what is gastrulation? Gastrulation is whereby the epiblast layer, not the hypoblast, the epiblast layer forms all 3 germ layers. Forms all 3 germ layers, which are what? The ectoderm, right? Ectoderm, the mesoderm, and the endoderm. All these germ layers come from the epiblast. the hypoblast would just disappear, right? Or some of the cells are found lining the yolk sac. Okay. So what we see is that, so this is the cranial part of the embryo, where you will see the, maybe just called the head part, right? And this is the caudal part, the tail part. So you see that from the midline going to the tail part, what you see is a streak, right? It's a streak, right? So when you say a streak, that's the point where we have an invagination, right? That is called the primitive streak, right? And at this point, what you see is the primitive node, right? The primitive node. So we see that cells of the epiblast, they invaginate downwards. So they actually dig down. So the cells, so if I have a hypoblast over there, and this is epiblast cells. The epiblast cells will use this streak, right? We use this streak, right, to invaginate and invade the other layers. So it actually first invade this hypoblast layer, invade the hypoblast layer. Right? And then when they invade the hypoblast layer, they form the endoderm, right? And also invade the middle part between the epiblast and the hypoblast, and then that form the mesoderm, right? So that is the point where the cells are, you know, just falling in, right? Right, and then first they invade the hypoblast, right? So they'll be falling in at this streak, and then when they fall in, they'll move in these directions. So they actually move in these cranial directions like that, cranial directions like that. Also move sideways, and they also move backwards, right? So I'm just illustrating that when they dig deep to that streak, when they fall into that streak, they'll move in these directions, right? That is the process of gastrulation, right? Okay, so as we see there, that is the epiblast, and then that's the primitive streak, that's the primitive node. From there, we create the notochord. Anyway, let's come to this point. So you see that within a streak, these cells are falling, then invading the middle part, and then invading the hypoblast, and then forming the endoderm. So they form the endoderm, in the middle part form the mesoderm, and then the cells that will actually remain form the ectoderm, right? Cells that actually remain then form the ectoderm, right? Fine. So that is what we call gastrulation. So we said in the process of digging deep, like here, when they move, they'll move in these directions. They move in these directions, right? And lateral and then caudal, right? Up to form now definitively at the end of the third week, these layers, right? So these are about peritoneal cavities, right? Anyway, but these layers. So what we see is that the ectoderm, right? The endoderm, and the mesoderm, but the mesoderm will then divide into compartments, all right? The mesoderm will then divide into compartments. So that's the neural tube. That is basically the neural tube that will come from the ectoderm. We'll talk about urulation just now. So the mesoderm will divide into a mesoderm that will be close to the notochord, right? So the notochord is basically a structure that will come from the endoderm, right? So the paraxial mesoderm, number one, paraaxial, so it's parallel to the axis, right? And then the intermediate is in between. And then lateral to that, we have the lateral plate mesoderm. And then we see that within the lateral plate mesoderm, we have a space that will actually form there. That is called the coelom. So this space will move laterally until it, you know, gets in contact with the extra-embryonic coelom as well, right? Extra-embryonic coelom. They just say mesoderm, but actually it's called extra-embryonic coelom, right? So this communication will cut the lateral plate mesoderm into a parietal layer and a visceral layer. So the parietal layer is the one that you see there in contact, that will be in contact with the ectoderm, and the visceral layer will be in contact with the endoderm, right? So that will create, so firstly, we say it will be like that, right? Having a cavity in between, and then the cavity will then start moving laterally until boom, like that. And then at the end, what I actually see is that we actually have two layers, right? The parietal layer of lateral plate mesoderm and then the visceral layer. The parietal layer will then form the body wall, and then the visceral layer will form the internal organs, right? The intermediate mesoderm, this intermediate mesoderm is going to form the urogenital system. That is going to form the urinary system, the kidneys, and the genital system, the gonads. So remember when we talked about the primordial germ cells migrating from the yolk sac to come and rest, right? It actually come to rest within the intermediate mesoderm and form the gonads, so the ovary and the testes. So anyway, here we have it. So the process of neurulation now, the ectoderm layer that will actually remain will form as well another groove, right? This groove, the neural groove, right? This groove. And in these cells over here that are called the neural crest cells that within the ectoderm, right? They approximate each other, right? Until we have closure of this groove to form a tube. So there we have a tube, right? So that is the neural tube that will form the brain and the spinal cord. And then these neural crest cells will actually migrate, right, from the tube and then come to lie on the lateral part of the tube and then form many structures within the body that will not be talked about in this video, right? But one of them is the dorsal root ganglion, right, of the spinal cord, the sympathetic ganglia, right, the suprarenal glands, right, and then also come to the ganglia, right, in association with the aorta, and we know they also go to the heart and also the face, right? These neural crest cells. So these are generally migrating mesenchyme cells. So those are the layers. The endoderm in yellow, the notochord there, right? And then the paraxial mesoderm, right? The intermediate mesoderm, right? The intermediate mesoderm over here, and then the lateral plate. So the lateral plate is now divided into a parietal layer that form the body wall, right? And the limbs, and then the visceral layer that form the internal organs. All right. So by folding, this disc will actually fold like that. That's called lateral folding. So what are the consequences of lateral folding? So the consequences of lateral folding is that this layer, so this part will become continuous with that part and then form one continuous tube. That is the tube. So that is the gut tube. So that's why we say that the endoderm layer forms the gut tube. All right? Yes. And here we have the parietal layer or the somatic layer, or what called the somatopleuric mesoderm, right? That form the body wall. So you see that's the body wall, and then that is the ectoderm that form the skin. Now makes sense, right? Right. And then around this gut tube is this mesoderm, that is the visceral mesoderm, right? The visceral mesoderm, right? Okay. And then that is the intermediate mesoderm over here that form the urogenital ridge, right? That will form the gonads and the kidneys. So now let's talk about what we get from these germ layers. So from ectoderm, we have the epidermis of the skin. So remember the epidermis of the skin. The dermis of the skin doesn't come from ectoderm, right? The central nervous system, the neural tube, the lens of the eye, the hair follicles, that's for the skin, the nails for the skin, the mammary glands, they also come from the ectoderm, as well as the sweat glands. So these are commonly asked within the exams, right? The hair follicles, the mammary glands, as well as the sweat glands. Then the mesoderm now being divided into three. The paraxial mesoderm will form the vertebra and the ribs. Remember we said that with the paraxial mesoderm, the intermediate mesoderm, and the lateral plate mesoderm that will actually split into two, right? So this, so we have the notochord in the middle, so we have the notochord in the middle, right? And then overlying the ectoderm, right? And then we have the endoderm over here. So we're saying that the paraxial mesoderm will form the vertebra and the ribs, muscles of the back, and the dermis of the skin. So in the exam, when they said the dermis of the skin comes from ectoderm, don't be fooled out, right? That is actually from mesoderm. Then the intermediate mesoderm will form the genital and the urinary system. That is that intermediate mesoderm. The lateral plate will form the heart, which is the splanchnic layer, right? Or the visceral layer. And then the visceral membranes, which is the visceral layer. But for the bones of the limbs and the muscles of the limbs comes from the parietal layer, right? And the endoderm forms the gut, the mucosa of the gut. Makes sense now, right? And then from the gut we have the liver, the pancreas, the gallbladder, the respiratory tract, the liver, right? That all comes from the gut. as well as the anterior lobe of the pituitary gland, right? The anterior lobe of the pituitary gland. And that finishes the discussion on the germ layers in the week 1, week 2, and week 3. Okay, week 1, week 2, and week 3. Okay,